members. I welcome you all, and I welcome Dr. Neelam Upadhyay, madam. Welcome, ma'am. Dr. Neelam Upadhyay. Dr. Neelam Upadhyay, ma'am, is graduated in food technology from Shahid Rajguru College of Applied Sciences for Women from University of Delhi in 2008. She completed her master degree in food science and technology from CCS Haryana Agriculture University in Hisar in 2010, and she did PhD in dairy chemistry from ICR and Dairy Karnal in 2014. Dr. Neelam has started her career by joining Agriculture Research Services as scientist in 2015. She served at Dairy Technology Division and Krishi Vigyan Kendra of ICR and Dairy Karnal in January till January 2023. Her research work in the area of detection of adulteration of foreign fat in ghee, development of functional foods, and extraction of natural colorants using green technology, formulation of their delivery systems, and application in dairy and non-dairy products. She has also developed a simple bilingual mobile app on scientific production of several processed commodities for rural youth and farm women, budding entrepreneurs, and new startups. She has joined IERI Institute, New Delhi in January 2023. She has also worked on DST-funded project as a PI with budget more than 30 lakhs. She has published more than 35 research papers, 4 review articles, 10 popular articles in various national and international journals. She has published around 12 book chapters, and she is the recipient of Best Paper Poster Awards from various scientific bodies, and she is the recipient of Young Woman Scientist Award. Research carried out under her dynamic guidance received Certificate of Excellence for extraction, characterization, and delivery of green carotenoids from carrot by waste for food applications with cash prize of 5 lakh rupees to the team by FSSI, Ministry of Health and Family Welfare. With this word, I welcome Dr. Neelam Upadhyay, ma'am, for her lecture. Over to you, ma'am.
Thank you so much, Anuradha, for the kind words. So I'll be today talking about the milk-derived bioactive peptides and their applications. Right, since I am told that the group also has some students, so I'll start from the basics and then we'll move on to the complexities, right? So proteins, they are the most abundant biological macromolecules which occur in cell. They show great variety, right? And with variety, I mean there are different kinds. Then they range from size of some amino acids to the polypeptides, and then they exhibit enormous diversity of biological functions. Since you all are aware that digestive enzymes like pepsin, trypsin, they are proteinaceous in nature. They are also the part of structural components like keratin. These days, keratin treatment of hair is also very popular. Then hormonal functions like insulin is the hormone, transportation, hemoglobin, then defense and protection and storage. So basically in proteins, we have amino acids as their monomer, or the building block of protein is amino acid, right? In amino acids, you can categorize the amino acids into different groups based on the R group. Okay, so here this is the structure of glycine, right? Where you have amino groups. See, the name is amino acids. So when I split amino acid, I have two words. One is amino and another one is acid. So which means that the structure should contain two things. One is amino. So here is the amino group, and then acid. So carboxylic acid is present, right? Then there is one R group which is there. And this R group varies from amino acid to amino acid. Okay. And then H is there to balance the valency of carbon. Okay. Glycine is considered to be the simplest amino acid, and it is achiral, whereas other amino acids are show chiral nature, right? non-polar aliphatic R groups. So based on that, you have like glycine, alanine, proline, valine, leucine, isoleucine, methionine. Then you have polar uncharged R group, which means now the R group will have like OH group. So serine, threonine, cysteine, asparagine is there. Then glutamine is there because of the presence of these groups, right? Then you have positively charged R groups. So lysine, arginine, histidine. Then negatively charged R groups, aspartate, glutamate, aromatic amino acids or aromatic R group, phenylalanine, tyrosine, tryptophan. These I'll be using in my subsequent slides. So it is like the basic that you must be aware of. This picture I have taken from Leninger. See, there is one more way of representation. So when I have to write a peptide chain, so either I can write a whole of this full name, or I can just use GLY, the three letters nomenclature, or I can just use a single letter nomenclature. Okay. So each amino acid is designated with this kind of nomenclature. You must remember this, especially the students, right? So as I said that amino acids are the building block of proteins, and they are joined by peptide bond. So what is this peptide bond? So here I have just given you a picture of 3 amino acids, okay, with say different side chain R1, R2, R3. Rest of the structure remains same. And the conventional way of writing the proteins are from N group, N terminal to C terminal, right? So here you see the first amino acid. So this is the amino group, then carboxyl group, amino group, carboxyl group, amino and carboxyl group. So here the OH from carboxyl group and H from amino group. So this moves out in the form of H2O, water, and then you get the formation of a peptide bond. Now here you can see that C double bond O, this group is aligned in one direction and NH group is aligned in another direction. And this is important when you from this primary structure of the protein you move to the secondary structure of protein. Say for example alpha helix. So this structure helps in the formation of the alpha helix so that the bondings can be formed, right? This O and the H of another amino. So and from NH, the H of another amino acid in the helix, the OH bond or the hydrogen bond can be formed, right? So this is the peptide bond, C double bond O and NH. This is the peptide bond, right? So this is the N terminal and C terminal. So in this, we have just the 3 amino acids. So it is tripeptide. Okay. Then from amino acid, we move on to the peptides, which is oligopeptide. That means different amino acids, they'll be arranged, they'll be linked by the peptide bond and you get peptides. Then from this, you get the tertiary structure, which means now the structure gets more complicated and the protein or the peptide, it tries to align itself based on the presence of the hydrophobic amino acids which are there or the hydrophilic amino acids which are there. So based on that, it will try to form a structure. Okay. And then is the quaternary structure, which means the 2 polypeptide chains, they are connected, okay, assembled together. So in this, if I talk about this picture, so this pink one could be one peptide and then one polypeptide, and this could be the yellow one could be the another one, right? So it is when the different polypeptides are there, then you get the quaternary structure of a protein. Now here you can note that in the primary structure here I have given the 3-letter nomenclature of each amino acid. Okay, this is just for representation purpose. Now I move on to the main topic, which is the bioactive peptides. So bioactive peptide, now I have just told you that what is there in a protein, okay, from say amino acid to basically the primary, secondary tertiary and the quaternary structure. But what are bioactive peptides? So here we are using the term peptide and we are not using the term protein, right? Why? Bioactive peptides, they are basically the specific protein fragments that have a positive impact on body function or conditions and may influence health. So here, as the name indicates, it is the specific protein fragment. That means not the whole protein, but a fragment or a portion of a protein will show the bioactivity or the positive influence or impact on the human body, and it will influence the health. So they are the organic substances, yes, because they are coming from proteins. Then they are formed by amino acids and joined by covalent bonds, encrypted in structure of a parent protein. So they are present in a parent protein, but then we have to cleave it or so that it can be released and then its function can be performed. So it is encrypted in the structure of parent protein and it is released or cleaved intact. Next is characteristics of bioactive peptides. So first, very important is that it is inactive within the sequence of parent protein, which means, as I told in my previous slide, that it has to be first cleaved, the protein has to be cleaved, and whatever peptide, bioactive peptide we want, that should come out of the whole protein sequence. Then only it will show its activity. So it is basically inactive when it is present in the parent molecule. How it can be released? Enzymatic hydrolysis by digestive enzymes, which is basically happening naturally in our body, wherein a lot of bioactive peptides are released after we intake food, for example, milk. Then proteolysis by enzymes derived from microorganisms or plants. So it can happen within the food system and also from food processing like cooking, fermentation, ripening, etc. Also, besides this, synthesis of bioactive peptides is also one of the ways of formulation of the bioactive peptides, which many of the companies they are using for formulating the bioactive foods in which they are adding, they are having the bioactive peptides. So they are synthesizing the peptides. Then another very important characteristics is that it contains 2 to 20 amino acid residues per molecule. Next important is it should not be digested in the gastrointestinal tract, which means that whatever amino acids are there in this, the whole sequence should remain intact and then it should move to the target site of action. Okay, suppose it is neuro, so it should move to that site. If it is pertaining to say another side, kidney, heart, liver, wherever, so it should move to that particular site in the body. Then they are absorbed through the gastrointestinal wall. Next is they act in the body as regulatory compound with a hormone-like activity. They have high tissue affinity, specificity, and efficiency in promoting health. So the tissues in our body, some ligands are there. So specific peptide will be binding to the specific site, and therefore it can show its action at that particular site. Some of it I'll take up in the subsequent slides. The sequence of amino acids of a particular peptide defines the function of peptide. Obviously, that what all amino acids are present in it, the composition will reflect the functions that the peptide can perform. And sometimes some peptides, they also show multifunctional properties, which means that they can show you different results, different outcomes from the same peptide. Okay, so for example, it can act as opioid peptide, and at the same time it can act as say immunomodulatory peptide, or it can act as the ACE inhibitory peptide or antihypertensive peptide. Source of bioactive peptides could be any plant, animal, fungi, microbes. So basically the proteins should be there, okay, and then the protein should contain the bioactive sequence that should be there. and then when hydrolysis is taking place, that particular sequence should come out intact, and then it can move out to the site of action in the body. So what are the various bioactive peptides? Opioid, antihypertensive, antithrombotic, then mineral binding, antioxidative, immunodefense. So I'll take up some of these. So bioactive peptides of milk origin, as the topic is given to me pertaining to milk. So the bioactive peptides of milk origin I'll talk about now. They exhibit various physiological activity. They show multifunctional properties, and sometimes they have presence of overlapping peptide sequence that exert different biological effects, right? So those who are not from the dairy background, milk protein contains two kinds of milk proteins are there in milk. So one is casein and another one is whey proteins, right? So if I give you a simple example, when you are making paneer, so the paneer which is obtained, it contains casein, and the water which is left out, which is also known as whey, it contains whey proteins. Fine. And further in this caseins, you have different proteins like beta, alpha, kappa, and in again whey proteins, you have different proteins like alpha-lactalbumin, beta-lactoglobulin, like that. So, for example, if I just talk about beta casein, so here I have marked few peptides or few fragments that are ranging from the 60 to 70 numbers, right? This decapeptide, then this is it contains 7, it is 11, and this is 7 amino acids. Then it contains 5 amino acids. It again contains 6 amino acids, right? So you can see that from 60 to 70, this particular range can show you the biological activity with respect to the opioid activity, ACE inhibitory, which means antihypertensive activity, immunomodulatory effect, right? But this will depend upon that what peptide is getting formed. So for example, if you have 60 to 70, so it is beta casomorphin 11, because 11 amino acids are there. Then 60 to 66, it is beta casomorphin 7, because 7 amino acids are there. Then this beta casomorphin 11 shows opioid activity, whereas beta casomorphin 7, it shows opioid, ACE inhibitory, immunomodulatory, all three activities together. Then you have from 60 to 64, this fragment, it is beta casomorphin 5. It shows opioid and ACE inhibitory activity. You can also see that all of these, they are obtained after the digestion with trypsin. Then we have immunopeptides, which can be formed from 63 to 68 amino acid fragment of beta casein. Okay. And also, like in this, I can also tell you about this A1 and A2, that concept, because that concept is related, A1, A2 milk concept is related to the beta casein. So in A2 milk, the at 67th position, you have proline. And in A1 milk, you have histidine at the 67th position. So when you have when you have histidine at the 67th position, so it can be cleaved, okay, and then you can obtain the beta casomorphin 7. So hopefully when this experiment was done by the scientists, so they might have taken the A1 milk, and as a result of which they are getting 60 to 66, this fragment. Okay. Next, now we'll move on to the specific peptides. First is the opioid peptide. So opioid, as the name indicates, it is the compound. Basically, opioid is a compound that resembles opium in addictive properties or physiological effects, pharmacological similarities to opium, which is morphine. It is named after Morpheus, the Greek god of dreams, and morphine is an alkaloid analgesic, which means it relieves pain, and it is isolated from opium. Okay, so this means that opioid peptides, they will be showing the effects of opium. And as I said, opium is obtained from, in opium, the active compound is morphine, and morphine is can be isolated from opium, like that. Okay, so in case of opioid peptides, we can divide them into two: atypical opioid peptide, typical opioid peptide, and atypical opioid peptide. Typical is like endogenous opioid peptide. Endogenous, whenever this term comes, endogenous means that they are produced within the body. Okay, and since they are produced within the body, so example is endorphins. Okay, then is atypical opioid peptide, exogenous opioid peptide. They are derived from the food. Exo means outside. So they are derived from the food, and generally the sequence contains 4 to 8 amino acids, and it is exorphins. Okay, because they are not of the origin from the body. Okay, they are derived basically from the foods. So opioid peptides, they are the opioid receptor ligands with agonistic or antagonistic activities. Means it can show the activity similar to opium or opposite to opium. The common structural feature of these peptides, here you can see that all these, so now here you can see that they are all of them, they are not from beta casein, unlike what I showed in the previous slides, right? Here, these are the different fragments from beta casein, alpha casein, and alpha lactalbumin. Okay. So, in the first one, you see 60 to 70, and in all these, you have tyrosine residue at the N terminal. I said no, that whenever we are writing the peptides, we write from N terminal to C terminal, right? So this happens to be the N terminal, and in this case, all these peptides, they have tyrosine at the N terminal, and also there is one significant feature or peculiarity of these peptides, that another aromatic residue, for example, phenylalanine or tyrosine, it is present at third or fourth position. So it can be present at the third or the fourth position. Here you can see the aromatic amino acid. Here you can see the aromatic amino acid. Here you can see at the third position. Okay. Then opioid receptors, I said in the beginning that they bind to the specific site of the action. So where their action is required, these peptides will go there and bind to the receptors which are present there. So opioid receptors, they are located in the nervous, endocrine, immune systems, and also the gastrointestinal tract of mammals. And wherever their action is required, they'll go and they'll bind there, and then they will carry out their actions there. The major and first opioid peptides discovered are beta casomorphins. They are the fragments of beta casein sequence between 60th and 70th residues, and have been characterized by new type of ligands. Next, we move on to the other milk-derived opioids. Like from alpha S1, you have casorphins. Then you have alpha lactorphin from human or bovine alpha lactalbumin, fragment 50 to 53. Beta lactorphins from bovine beta lactoglobulin 102 to 105. Then also some of the opioid antagonists are there. They are generally found from the kappa casein. They are known as casoxins. And here are some examples like casoxin 6, casoxin C. Okay, and the sequence is this. Next, we move on to antihypertensive peptides. So as the name indicates, they will be helping in reducing the blood pressure. Okay, and how it happens is, so see, this when we talk about blood pressure, so it is a general mechanism is renin angiotensin system. Okay, this more about this, if you want to know, you'll have to go back to your 11th 12th classes and read about the excretory system in mammals or humans. Okay, here so based on that, the blood pressure will be controlled. Okay, but now how these peptides can be helpful in uh, playing the antihypertensive role? Okay, how? See, angiotensinogen. It is there in the body. Okay, and it is in the inactive form. Fine. Then renin is what we say. What happens is, so uh, if I talk about kidney, so when blood volume is low, then there are some cells in the kidney known as juxtaglomerular cells in the kidney. They will secrete this renin directly into the circulation, right? And the plasma renin then carries the conversion of this angiotensinogen, which is released by the liver, and it is converted into angiotensin 1. Okay, so this contains more than 400 amino acids are there. So whenever you have this term, no, angiotensinogen, oh, this term is there, no, then this reflects the inactivity of a protein, you can say, or the hormone, you can say. And when this ogen term is removed, then this is converted into an active form. Okay, so here in this case, renin converts the angiotensinogen to angiotensin 1, which is basically the decapeptide, and it is from the N terminal. So first 10 amino acids from the N terminal, they are cleaved and they lead to the formation of angiotensin 1. Okay, this is a decapeptide. Then on this angiotensin 1 converting enzyme, or commonly referred to as ACE, ACE, angiotensin 1 converting enzyme, is also known as ACE, angiotensin converting enzyme. So this enzyme converts angiotensin 1 into angiotensin 2. Okay, and this angiotensin 2, as soon as it is formed, so it is a vasoconstrictor. Fine. As I said, that when the blood volume is low, so which means that the pressure will be low. So at that time, we need to increase the pressure, right? So in such case, angiotensin 2 will be helpful because it will constrict the vessels, as a result of which pressure will be increased. Fine. Then it in normal human beings, right? One more thing is there, this angiotensin 1 is converted into angiotensin 2 by removal of dipeptides from the C terminal. Again, HL. I told you, that one letter nomenclature is also can be given to the amino acids. So I have used that here. So now see, this ACE is very important here because it is involved in the formation of a vasoconstrictor. Next thing what it does is, there is bradykinin. Again, it is present in the body. So it is a vasodilator. Okay, vasodilator means it will reduce the blood pressure. Okay, so angiotensin 1 converting enzyme, it converts this into an inactive peptide, which means that if somehow I inactivate this angiotensin 1 converting enzyme, then what will happen? This vasoconstrictor will not be formed, as a result of which the pressure will not be increased, blood pressure will not be increased, and also since this enzyme is now inactive, so vasodilator will be active, as a result of which we are creating the conditions which are antihypertensive, or which decrease the blood pressure, which do not lead to increase in the blood pressure, right? So basically, the antihypertensive peptides, they will inactivate, or they basically inhibit the action of angiotensin 1 converting enzymes. Okay. As a result of which angiotensin 2, it is inhibited, and also bradykinin, which is there, it is activated, right? And thus hypotensive effect will be shown. Now, what are the various peptides, ACE inhibitory peptides? So ACE inhibitors derived from milk proteins, casokinin. So in the opioid ones, we were using the term orphins, okay, like casomorphins, like that. And here we are using the term kinins. Okay, so casokinin, it is a fragment of casein, and these peptides, they show ACE inhibitory activities. For example, beta casein 193,202, beta casokinin 10, because 10 amino acids are there. It shows ACE inhibitory and immunomodulatory effect. Alpha S1. So here we are using the term casokinins. So alpha S1, casokinin 5, because it is derived from alpha S1 and it has 5 amino acids. Now here, there are several researches which say that the ACE inhibitory peptides, they contain this proline as an important amino acid. Okay, so this valine, proline, isoleucine, proline, and isoleucine, proline. These fragments, they were separated from, or they were purified from, Japanese soft drink, which is known as scalpis, and it is obtained by fermentation of the skim milk by Lactobacillus helveticus and Saccharomyces cerevisiae, right? So from this soft drink, they isolated the VPP and IPP, and they said that it possesses ACE inhibitory activities, right? There is also one more thing associated with this, that angiotensin 1 decapeptide, when it is converted to angiotensin 1 converting enzyme, so this angiotensin, when it is acted upon by ACE, then angiotensin 2 is formed, and this also has an effect on the bones. It decreases the osteoblast differentiation and increases osteoclast resorption. So what does this mean? If you have decreased osteoblast differentiation, this means now the bone forming activity will be less. Okay, and bone breaking activity in the body will be more, which means again it is showing a negative effect on the bones. Fine. And therefore ACE inhibitory activities, if they are ACE inhibitory peptides are there, then they are going to show the positive impact on the blood pressure and also on the bone health. So some of the studies have reported this also, this correlation also they have reported. Next comes is anti-thrombotic peptides. So thrombosis, we know that when like blood clot is formed, that is thrombosis. Okay, anti-thrombotic means these peptides, they are showing the negative effect to, like they'll not promote the formation of thrombosis. Okay, and in some conditions it is required that the blood clot should not be formed. So how these anti-thrombotic peptides, they are there in the milk, and how they're correlated to the thrombosis process which is taking place in the blood. So clotting of milk involves interaction of kappa casein with chymosin, we know. Then clotting of blood involves the interaction of fibrinogen with thrombin, and these mechanisms, they are remarkably similar. So the red ones I have written are for the milk, and the black ones, they are happening in the blood. So prothrombin is converted into thrombin. Then fibrinogen, this thrombin converts the fibrinogen to fibrin plus fibrinopeptides, and this fibrin leads to clotting of blood in case of milk. So kappa casein is acted upon by chymosin, as a result of which para kappa casein is formed and glycomacropeptides are formed. Para kappa casein is cheese, basically cheese, right? It is basically a clot or cheese in the form of cheese, and then we have glycomacropeptides which are released. Fine. So this is a similarity between blood and the milk, and this probably this is the reason why some of the milk derived peptides, they show anti-thrombotic effect. So main anti-thrombotic peptide is isolated from bovine capacasein, casopletlin. So here we will use the term pletlin is used for showing the anti-thrombotic activities. Casopletlin, they inhibit the aggregation of platelets and binding of fibrinogen to platelets, as a result of which they will prevent the formation of the blood clot or thrombosis. Structural homology exists between this casopletlin and human fibrinogen gamma chain. Then human milk derived antithrombotic peptides. This is structurally similar to the fibrinogen alpha chain, and it inhibits platelet aggregation, thus acting as antithrombotic in nature. Next comes as mineral binding peptide. So here in mineral binding peptides, it is important phosphopeptides. They are mostly phosphopeptides. Why? Because phosphate group will have negative charge, right? And if negative charge is there, that means some positive minerals can bind to it, as a result of which the peptide can show the mineral binding effects. So phosphopeptides can form soluble organophosphate salts, and they may function as carrier for different minerals. The high concentration of negative charges of phosphate peptides make them resistant to further proteolysis. The negative charged side chain, that is phosphate group of amino acid, it represents the binding site for minerals. Casinophosphopeptides, CPP, they are the large group of peptides that have a phosphoryl residue in common. This I have already said. So these peptides are formed either from casein by proteolytic enzymes during fermentation or in GI tract. Then they can form soluble organophosphate salts and lead to enhanced calcium absorption by limiting the precipitation of calcium in the distal ileum. And this I have said because of the presence of negative charge basically on the phosphate group. It can bind to the positive minerals like calcium, magnesium, iron, zinc, and CPP can bind and solubilize minerals, physiologically beneficial in prevention of osteoporosis, dental caries, and anemia. Obviously, because if the content or intake of or absorption of calcium and iron, zinc, all these things, they are more, then these problems can be reduced. It is also said that calcium binding phosphopeptides, they show anti-karyogenic effect. Anti-karyogenic effect means the carie, which is the dental caries, will not take place. And how? Because inhibition of caries lesion through recalcification of dental enamel. It is suggested that these peptides can also lead to recalcification of dental enamel, as a result of which they show anti-karyogenic effects. Then anti-karyogenic effect may also be related to the ability of CPP to compete with the dental plaque forming bacteria like Streptococcus mutans for calcium, their application in the treatment of dental disease. So some people, they have also formulated some bioactive peptides containing toothpaste. Okay. So these are the examples. Mineral binding activities: alpha S1 casinophosphate, alpha S1 casinophosphate fragments are different in this: 43 to 58, 59 to 79. Then beta casinophosphates, fragment 1 to 25 of beta caseins. Next is the bioactive peptides which are found in market. So as I said, calpis, this I already talked about. It is the sour milk, or we can say fermented milk. Okay, it contains VPP, IPP. Then it shows antihypertensive activity. It is in the form of drink. Then evolus, again the source is milk. It also shows antihypertensive activities. VPP, IPP is there. Valine, proline, isoleucine, proline is there. Then lacprodan is there, in which the source is whey proteins. It regulates the blood sugar level. It is in the form of powders. Then bonitopeptide. Bonito is coming from fish. Then we have lecithin, which comes from milk. It contains the amino acid sequence this. It is considered to show stress relief properties. Then we have capolec. Like there are several. From soya, again, we have some cholesterol blocks. It act as, the brand name is cholesterol block of the company, right? Then we have, they show the cholesterol reduction activity. It is in the form of drinks, right? And there are several infant formulas also now coming up, which say that they contain these particular bioactive peptides, and a particular beneficial effect is shown by these bioactive peptides. So these are some of the references that I have used. And thank you. Now I think we can take up some of the queries if you have any.
Thank you so much, Dr. Neelam Upadhyay, ma'am, for nice and informative session. Now the session is open for the discussion. Participants, if you have any query, please ask.
Students, if you have any query, you can ask me.
I think, ma'am, there is no query. Okay. You have elaborated your topic in a very informative way and simple way. So, on behalf of organizing committee, I would like to thank Dr. Neelam Upadhyay, ma'am, for this nice presentation. Thank you so much, ma'am. And thank you all the participants for your active participation. Thank you, ma'am.
Thank you, Dr. Anuradha.
Okay, ma'am. Ma'am, you can leave now.
Okay, fine. Thank you.